Okay. Hello and welcome. Thank you for joining us for the fourth annual Black in Immuno Week, a week where we will celebrate, amplify, and support Black voices in immunology. Before we begin, we would like to thank our major sponsors who have made this week possible. You can find a full list of sponsors on our website. Don't forget to tweet about our session today using the hashtag AutoImmunity Allergies with each word capitalized for accessibility purposes. We will have live captions and ASL interpreters during the sessions. Click on the CC at the bottom of the screen to access captions. You can submit questions through the comment section on the platform, not the Q&A. As a reminder, we require all audience members to be polite and respectful while in the Black and Immuno space. If we see anything offensive, your comment will be deleted immediately and you will be removed from the platform. After the conclusion of the week, recorded videos will be posted on our YouTube channel and transcripts will be made available on our website, blackinimmuno.org. So my name is Yasin Al-Faki, and I'm part of the Black in Immuno planning team. And I will be the moderator for our Science Talk session today, Autoimmunity and Allergies Science Talk. We have 4 speakers here to tell us about their work. They have each about 15 minutes for their presentations, and we will follow with a 5-minute Q&A. Without further ado, let's get to it. So our first speaker for this session is Dr. Elaine Kouame. Elaine is currently a postdoctoral researcher with Dr. Joanna Harkles and Dr. Gabriella Fragitilis at UCSF, focusing on uncovering the prenatal factors that shape early human immune development. Integrating wet lab expertise with high-dimensional computational analysis, she aims to advance our understanding of early life immunity and its lasting impact. Before her postdoctoral work, Elaine pursued her PhD in immunology at the University of Chicago, investigating the mechanisms by which enteric viruses trigger immunopathology, especially in the context of an autoimmune-like disorder, celiac disease. Along her scientific endeavors, she has actively contributed to fostering inclusivity within the University of Chicago's Biological Sciences Division and led the STEM initiatives in underrepresented communities. Elaine, welcome, and we're excited to see your talk. Hear you talk.
Amazing. Thank you so much, Yasin, for the introduction. I'm very honored to be here today. This is my first time presenting at Black in Immuno after being a member and participating every year. So I'm truly honored to finally share a little bit of my science. So without further ado, I'm going to start. Awesome. Okay, you can see my slide fine, correct? Awesome. Okay, so today I'm going to share with you some of my PhD work where we focus on understanding mechanism by which viruses induce loss of tolerance to dietary antigens. So one of the main questions of the immune system is basically how does the immune system respond to foreign but harmless antigen that are found in the gut. So I haven't had breakfast this morning, but I'm pretty sure like some of you had breakfast this morning, and we eat food on a daily basis. So which means that what it means for the gut is that it's about 130 and 190 grams of protein that's been absorbed in the intestine daily, and this is
Elaine,
potential. Mm-hmm. Sorry, what's up? Can you hear me? Oh, okay. I can see the. Okay, let me try again. Good. Thank you. Okay. Thank you. I somehow. Okay. Yeah. Okay. I'm gonna start over then. Can you see? Can you tell me? Because I can't see the screen actually. Can you verbally let me know if you can see my slide? Okay, I will take it as a yes. Okay. So what I was saying is that For my PhD project, I was interested in studying mechanism by which viruses can disrupt the immune response against dietary antigens. And the slide I was explaining earlier is that one of the challenges of the immune system is to be able to respond adequately against food antigen. And there's about 130 and 190 grams of protein that is being absorbed daily in the small intestine. The question is, how do immune cells respond to something that is foreign but harmless? Studies have shown that tolerance to dietary antigen in humans is mediated by T cells, and there have been studies in some of my things done. Okay, there have been studies in mice. In this study here that I'm showing, where they compared mice, germ-free mice that were weaned either on the regular diet that has dietary antigen, dietary proteins, or a diet with no antigen and no proteins in the dark dot. So you can see that mice that were weaned under a child diet with dietary antigen, you can see the induction of regulatory T cells in the guts. And by using a very specific system where you can track the T cell that's specific for a dietary antigen, and in this model they use ovalbumin. So you can transfer T cell that's specific to ovalbumin, feed mice with ovalbumin, and look at the expansion of these T cells, and look at what type of T cells these cells are. So you can see that upon 5 days post-feedings or 12 days post-feeding, you can see this expansion of regulatory T cells in the gut, really suggesting that the way the immune system deals with this foreign but harmless antigen is by inducing these regulatory T cells. And we do know that failure to induce tolerance against dietary antigens can lead to food allergy and celiac disease. So for my PhD work, I was interested in understanding the mechanism that lead to the induction of celiac disease. So what is celiac disease? So celiac disease is kind of like an imposter in the autoimmune field. It's an autoimmune-like disorder that is driven by dietary gluten. So genetically predisposed individual will lose a tolerance to gluten, and which will lead to autoimmune-like symptoms. So how does this work? Upon ingestion of gluten, you have differentiation of Th1 T cells that are specific to gluten. These are inflammatory T cells. These cells can license plasma cells to secrete autoantibodies. So they will secrete antibodies against gluten, but also antibodies against self-antigen. These T cells can also license CD8 lymphocyte in the gut, and these can go and then induce epithelial cell killing and leading to villus atrophy. So this puts the gluten-specific Th1 T cells at the center of celiac disease. So they are the main driver of all the pathology that you can see in celiac disease upon ingestion of gluten. So this is a little bit different from a classical allergy model, because here you have a foreign antigen, which is gluten, that is driving this autoimmune response. So for my PhD project, I was interested in understanding what are the events that lead to this Th1 T cell response. So how does gluten introduce this Th1 T cell response instead of what you expect as a regulatory T cell response? So we look in the literature. Something that we found very interesting is that although genetically predisposed individuals that can develop celiac disease, those are individuals that express HLA-DQ2 and HLA-DQ8, which are the MHC molecule that can bind to gluten. And you can see that with this graph that only a fraction of these individuals develop celiac disease. So this suggesting that genetic factors alone cannot explain the disease. And there have been studies in humans showing correlation between rotavirus infection and an increased risk of celiac disease in children. Interestingly, patients also affected by chronic hepatitis C that receive type 1 interferon treatment also started to develop celiac disease. So suggesting that viruses or viral factors can drive the celiac disease pathology. So the question we asked is, how do viruses change the immune response against dietary antigen? To answer this question, we use a viral model. So we use reovirus. Why reovirus is a very tractable system and very easy to manipulate in the lab. So reovirus, it is a double-stranded RNA virus that has 10 genes. It's a very small virus. Yes, those genes are segmented, so it's very 10 pieces of genes. They can be segregated based on size. So you have the large genes on top, the medium genes, and then the small genes. The site of entry for reovirus is Peyer's patches. So these viruses are enteric, so meaning that it can affect the guts. One thing that's also very interesting about celiac disease is that about reovirus is that it can affect human and mice. And then we've shown previously that celiac disease patients have a higher viral load or viral antibody titer as opposed to non-celiac disease individual, suggesting that there's a correlation with this increase of celiac disease antibodies in people affected by celiac disease. Okay, so what we wanted to have is having two viruses that can from the same family that can both infect the gut, and hopefully one of these viruses will be able to disrupt the immune response to dietary antigen, and we can understand why, and the other could be used as a negative control. So to have that, we genetically modify reovirus. So here I'm showing two strains of reovirus, so T1L strain and the T3D-RV strain. So what we did, we induced two of the genes that are required for infecting the guts into a T3D virus. So then we are in the system where we have two viruses that can both infect the gut. So we show that both viruses can replicate in the gut. They can induce a protective immune response, so they are able to stimulate the immune system. We also wanted to have a system that we can track the T cell response against dietary antigen. So we used immunologist's favorite model, so this OT2 T cells. So in this model, in this presentation, what I will show you today, we will transfer OT2 T cells. We infect mice with reovirus and then put them on an ovalbumin diet. And we'll be looking at the T cell response in the mesenteric lymph node, which is the site where the immune response to food antigen is being mounted, and the lamina propria, where we can also observe the effector response against dietary antigen. So the lab has shown previously that we have T1L and T2D-RV, so those two viruses that can infect the guts, and we show that only one virus was able to induce regulatory T cell response. So only one virus was unable to disrupt the immune response to food. So here I'm showing you flow cytometry results of those T cells that will transfer post-infection and post-antigen feeding. So you can see that upon infection with T1L, you lose this FOXP3 Treg response that you normally observe in sham, and you do not see a striking effect with this T3D-RV virus. What was interesting here, similar to celiac disease, with infection with T1L, you now develop a Th1 response, an inflammatory T cell response against dietary antigen, ovalbumin in this case, and you do not see the same effect with T3D-RV. So now we are in the game. We have a system where we have one virus that can drive Th1 response against dietary antigens, similar to what we observe in celiac disease. So for my project, I was really focusing on 3 questions, and today I'm going to show only one of those questions. So we are interested in the main question, which is, what are the T1L host interactions that are leading to Th1 immunity against dietary antigen and this loss of tolerance to dietary antigens? So today I'll be focusing on what are the host molecular and cellular factors that are required. So before I go to that, I want to introduce dendritic cells. So dendritic cells have been described in the literature are very key in mounting this regulatory T cell response, especially dendritic cell DC1. So dendritic cell can be divided into 2 categories. Can be either resident or migratory, and they have they differ in the ability to induce tolerance, in this case, regulatory T cell response. And DC1 has been shown to have the highest ability to induce immune response against dietary antigen, so tolerance. So For my project, I was interested in investigating whether these DC1 also play a role in this disruption of the T cell response against dietary antigens. So to answer this question, we use DC1 knockout mice. So these mice lack the transcription factor that's required for generating DC1. So these are BADF3 mice. So they're BADF3 knockout. They lack DC1, but they do have the other subset of DCs, those DC2s. So you can see here with flow cytometry data, so we transfer those T cells, infected them with T1L. We have wild type mice control, and then the BATF3 knockout mice that lack DC1. You can see that we lose this induction of Th1 response. So suggesting that migratory DCs are required for this disruption of T cell response against dietary antigen. So how do they do it? So we first look at viral titer. So maybe the virus is not able to replicate. You can argue that the virus is not able to replicate if those DCs are not there, but that wasn't the case because we didn't see any disruption in viral replication. So there was no difference between wild type and knockout in the amount of virus. And there's also there was a delay in the type 1 interferon response against the virus, but this was quickly recovered in the mesenteric lymph nodes. So there doesn't seem to be any defect in viral sensing or in the response to the virus. So what is the mechanism then by which DCs are driving this aberrant response against dietary antigens? So we know DCs, dendritic cells, are very good at mounting, at presenting antigen to T cells. So we ask, but maybe it is because dendritic cells, those DC1 are the only cells that can present dietary antigen. So we generated a mouse model where we can delete this MHC molecule from the DC1 subset, and we then assess the Th1 response. So by deleting this, the MHC molecule from the DC1, we can see a reduction in the Th1 response, but it wasn't completely gone. So this can suggest like a partial effect, but also that there's still antigen presentation happening in the system. But it could also be an experimental issue where we are not able to delete completely the MHC molecule. So we see a partial effect in conclusion, I would say. Okay, so then we are also interested in something that is another factor that is unique to the dendritic cell that can explain this phenotype. We know from the literature that dendritic cells express this molecule called CLEC9A, and this CLEC9A is a C-type lectin receptor that recognize necrotic cells. We knew from the literature that reovirus C1L can induce cell death and necroptotic type of cell death. So we curious to see maybe the reason why those dendritic cells are able to sense, are able to drive the Th1 response could be because they are able to sense cells that are dead due to viral infections. So we use a blocking antibody to block CLEC9A, and then we can see that also a significant reduction of the Th1 response upon blocking CLEC9A, suggesting that CLEC9A sensing of these dead cells might be important for generating this Th1 response. Lastly, now we have this phenotype with sensing dead cells. So we wanted to investigate whether cell death actually played a role in this system. So just to orient you, so we are looking at necroptosis, which is an inflammatory type of cell death where you have a cascade of events leading to activation of MLKL, and this factor will basically poke holes in the cells and release intracellular factors, and this will kind of have a cascade of inflammation. So this has been associated, this type of cell death has been associated with autoimmunity and also in the context of anti-tumor immune response. So we generated, we got this MLKL knockout mice. So these mice cannot undergo necroptosis upon viral infection, and we saw kind of a similar phenotype. We saw like this reduction in this Th1 response here, really suggesting that the ability of the virus to kill the cell and being sensed by the dendritic cell is key to mount this aberrant response against dietary antigen. So in conclusion, we have this model where at homeostasis, what you would expect in the context of food antigens, you will expect that these antigens will reach out the gut, they'll be picked up by dendritic cells, in the same system, there will also be apoptotic cell death because there's constant recycling of epithelial cells in the gut. And this, the dendritic cell will kind of incorporate these environmental factors and then migrate to the lymph node and induces regulatory T cell, and that's what you see at homeostasis. However, what we found in our research is that upon viral infection, you now have a disruption in the system where you change the type of cell death that you have. You have necroptotic cells here. So now the dendritic cells are not only uptaking food antigen, but now they are integrating a different kind of cell death. And this, they are integrating this through CLIC9A. And I didn't show this data today, but we also see that those dendritic cells are the main producers of type 1 interferon and IL-12. And these factors, especially type 1 interferon, is necessary to prevent the regulatory T cell response. And then together, they drive this Th1 response that you see. Okay. So with that, I would like to thank my lab, my PhD lab. I really had a great time in my lab. I spent 6 amazing years in Chicago, even though it was super cold. And also want to thank our collaborators, the Dermody lab, the Sterility lab, the Rosenfield lab, and the Barrero lab, and again, the Committee of Immunology at the University of Chicago. With that, I will take question. Thank you so much for your time.
Great. Thank you very much. That was really, really nice and really, really fantastic. Amazing work.
Thank you.
Yeah, while we wait for questions, I may ask actually a few of mine. So just to use the fact that I am chairing this. So basically, I also saw in your models, especially when you like genetically ablate the CDC ones, that you have a reduction in TBET positive Tregs, and I know that those are like important for suppressing Th1 responses. So
Yeah.
are we talking here about a cause and effect, or are we talking about something that is, I mean, are they decreased because there is an overwhelming Th1 response, or are they first decreased and this enables Th1 responses to be like exaggerated?
Yeah, that's very interesting questions, like how those two T cell responses are connected, because you will think that the Treg response are normally induced to prevent the Th1 response. So in this case, yeah. So let me take a step back. So we have the DC1s. When we remove the DC1, we completely lose the Th1. So that we can see the effect. The effect is more drastic in the lamina propria. So we think that some cells are induced, but they just don't stay. So when you look later on in the lamina propria, you see those Th1. So we know that dendritic cell DC1 are key. So other DCs cannot induce this phenotype. What is interesting is that because we see a blockade of the regulatory T cells, so the absence of these T cells, even though the DC ones are not there, suggesting that other DCs can actually regulate the T cell response, but only the DC1 can induce the Th1. So it could be like the DC1 and other DCs can prevent this regulatory T cell response, which we think is due to type 1 interferon. So all the cells that are taking viral particle or viral infected cell maybe can secrete type 1 interferon. And then we know, I didn't show that today, but we know that type 1 interferon is needed to block the Tregs. But because DC1s are unique in their ability to first sense the dead cells, but also secrete IL-12. So they are unique in their ability to induce the Th1. So it's kind of like a division of labor when you have like a subset of cells that can induce the Treg, and then only DC1 can drive this Th1. So we kind of need both.
Oh, okay, cool. Thank you. So we have a question actually from our next speaker. He said if you see a decrease in like Tregs in general, not the Tibet positive ones only.
Yeah, yes, I do. Like just for the sake of simplicity, I didn't add those data, but all the Th1 response were associated with the absence of a regulatory T cell, like I showed you in the beginning, with the beginning of the model with T1L infection. So we know that when you have T1L infection, you induce the Th1 and you block the Tregs. So we still see this blocked Treg in the absence of DC1, in the absence of CLEC9A, like I was saying, that because we think that other cells can, in response to the virus, other cells can drive this blockade.
Okay, thank you. One last question, then we move on. It's from me actually. Can you actually model gluten in your infection model as you take ova out and you put gluten? And I don't know also if you can, if the mice can get sensitivity to gluten, or if you can also have TCR
Yeah.
transgenic mice for that.
Yeah, that's a good question. So my lab generated transgenic mice that have this human HLA, because
Mm.
the main issue is having the correct HLA to respond to gluten, and mice do not have that HLA. So we generated an HLA DQ2 and DQ8 mice, and then we can give them gliadin, which is the component of gluten that the T cell are responding to. And then we can see that, yes, upon T1L infection, we'll see this Th1 response. But the thing that we're not able to recapitulate in this model is the Treg part. We do not see like a Treg induction upon
Mhm.
just feeding gliadin, so we think might be an artifact of the system, but like we're unable to not we are not able to reproduce this part of the phenotype. But at least the Th1 part still stays the same, even with a different model.
Cool, cool. I think we have also like time for two more questions. So there's a
Okay.
question from Nzinga Mac. She says, "Great work and great presentation, Dr. Elaine. You mentioned the computational aspect, if I'm not mistaken. What are you modeling computationally?"
Okay, yeah. So I'm not modeling computationally per se here. So what we've done here, we sequenced the different DC subsets and trying to see whether transcriptionally we can find a program that is associated with the Th1 response, but also we can find a unifying program that is associated with the regulatory T cell response. So we try to, at the transcriptional level, to see how we can decipher what program correlate with each type of response. But I'm not doing any modeling with this project. I think maybe you're referring to my current postdoc work, where I'm doing modeling in the context of fetal immunity, but that's like not the session. But yeah,
Or
really good question though. Yeah, good question too so far. Yeah.
Yeah. The last question is actually from my postdoc supervisor, Professor Lucy Walker. She said, "Fascinating talk, thank you. Have you looked at other models of virus-induced pathology in the MLKL knockout mice, like
That's
models
a
of virus-induced autoimmunity? How generalizable is the need for necroptosis?"
Yeah, that's a really good point. The short answer is no. We haven't looked at other viruses in specifically in the absence of cell death, but that's a current project from a PhD student in the lab. Is He's currently testing different viruses with this MLKL system, but we've tried norovirus on top of this virus, but in the wild type system. So we try the chronic strain of norovirus CW3, and then the so what's the opposite of chronic? The transient strain.
Acute. Acute.
So acute, thank you. And the acute strain, and we saw that the acute strain had an effect, also was able to drive Th1, but it's a different model system, because this norovirus infect most like APC, while reovirus infect more on the epithelial cell side. But we don't know in that system whether cell death plays a role as well. But it's a really good question, and we are currently following up on that to see.
Cool, cool. Thank you very much, Elaine. Thank you very much for your talk and the discussion. And I think we now move on to our next speaker.
Awesome. Thanks for having me.
Thank you. So, our next speaker for this session is Dr. Prince Baffour Tonto. Prince Baffour Tonto was born and bred in Ghana. He finished his undergraduate studies in biochemistry at the University of Ghana. Then he was merited with the Japanese government scholarship to pursue his master's and PhD at Mi University in Japan. He is currently doing his postdoctoral fellowship at Rutgers University in New Jersey, where he is studying the cellular immune responses to dengue fever and yellow fever viral infections. So we are excited to have you, Prince, and yeah, take it away. Thank you. I think you are on mute, Prince. Oh, yeah.
Okay, can you hear me?
Yeah, now we can hear you.
All right, thank you very much, Yasin, for this opportunity to present my work at the Black Immuno Week. Let's see.
If you can go to your first slide and then go into presenter mode. You start the slideshow.
Yes, I've done that, but let me see. The present. Okay.
It seems like it's frozen.
Yeah.
Probably could try unsharing and then sharing again. That would solve it.
Okay. Okay, so today I'll be talking about influenza vaccine allergy, specifically.
We don't see the slides yet. Now we see it. Okay, thank you very much.
Okay, so I'll be talking about influenza vaccine allergy, specifically the mechanism by which the influenza vaccine induces allergic reaction to persons after receiving the influenza vaccine. Okay, so immunization is an important public health intervention that is being done to reduce the disease burden caused by infectious pathogens. But then adverse events that follows immunization continues to hamper future immunization programs. And these adverse events are categorized into minor events and severe events. And examples of minor events are headache, fatigue, fever, chills, myalgia, injection site pain, and swelling. An example of severe adverse events are convulsion, encephalitis, vasculitis, Bell's palsy, anaphylaxis, which is the central focus of this presentation, and Guillain-Barré syndrome and thrombocytopenia. And the components of vaccines that are being implicated to induce allergic reactions includes egg proteins, formaldehyde, gelatin, neomycin, polyethylene glycol, polysorbate, and thimerosal. And anaphylaxis is the most severe form of an IgE-mediated allergic reaction. And it affects multiple organ systems in the body. And the most notable systems are the respiratory system, the gastrointestinal system, the skin, and the cardiovascular system. And so there is a model showing the cellular and molecular mechanism of an IgE-mediated allergic reaction. And so basically, when the body becomes exposed to an allergen, dendritic cells process the allergen and induce the activation and differentiation of naive T cells to Th2 cells. And Th2 cells releases a signature cytokines, which is interleukin 4 and interleukin 13. These cytokines induces antibody isotype class switching in B cells to the production of IgE antibodies. Then the IgE antibodies then binds to high affinity receptors on mast cells or basophils. And this is known as the sensitization phase. And so subsequent exposure to the same allergen cross-links the IgE that is bound to the high affinity receptors on the mast cells or basophils to induce the activation and degranulation of these cells to release vasoactive amines and lipid mediators of inflammation. And these causes hyperpermeability of the blood vessels and also smooth muscle contraction, leading to the clinical manifestation of anaphylaxis. And anaphylaxis is a rare event that follows influenza vaccination. And so the seasonal influenza vaccines, like the bulk of the seasonal influenza vaccines are produced using eggs. Okay, so the inactivated influenza vaccine contains traces of ovalbumin, which is the major component of egg white proteins. And so before, like people thought the ovalbumin was the cause of anaphylaxis from influenza vaccination. But then influenza vaccines, which are produced using recombinant hemagglutinin protein, also causes anaphylaxis. And these recombinant influenza vaccines are devoid of egg proteins. So the so then the question was, what component of the influenza vaccine mediates influenza vaccine associated anaphylaxis? So since anaphylaxis is an IgE mediated allergic reaction, we first determine the influenza vaccine specific IgE levels in patients who develop anaphylaxis, and also in patients with previous egg allergy who received the influenza vaccine, and also healthy subjects who also received the influenza vaccine. And we observed that the patients who develop anaphylaxis after vaccination elicited high IgE responses to the vaccine. And we also observed that the hemagglutinin protein, which is the active ingredient in the influenza vaccine, elicited high IgE responses in patients, like in the patients. And so we concluded that the hemagglutinin protein is the cause of anaphylaxis. So we concluded that the hemagglutinin protein is the allergen that induces allergic reaction to the vaccine. And so to further shed light on the pathogenesis of the influenza vaccine associated anaphylaxis, we analyzed the influenza vaccine specific IgE levels in children aged from 0 to 18 years, and we categorized these um, subjects. So like these were healthy subjects. So like we administered these subjects with 2 doses of the influenza vaccine. Yeah, but before that, we categorized these subjects into 4 age groups. That is 6 months to 2 years, 3 to 5 years, 6 to 12 years, and 13 to 18 years. And each subject received 2 doses of the influenza vaccine. And we observed that the subjects in the younger age groups had increase in influenza vaccine specific IgE levels after vaccination. Yeah, but none of the subjects experienced anaphylaxis after receiving the influenza vaccine. So then we were curious to find out why none of the subjects experienced anaphylaxis. So okay, so this is a model um, showing how IgG4. So IgG4 is an anti-inflammatory molecule that inhibits allergic reaction, and it does that by competing with IgE to the binding of allergens. Right. So we then investigated the. So we then evaluated the influenza vaccine specific IgG4 levels in the subjects who were sensitized to the vaccine as compared to subjects who were not sensitized to the vaccine. And we observed that the subjects who were sensitized to the vaccine had increased levels of specific IgG4 as compared to their non-sensitized counterparts. And so we then evaluated the specific IgG4 IgE ratio in these subjects after vaccination, and we observed that the specific IgG4 IgE ratio increased significantly after vaccination in all the age groups. And so these data suggest that even though the influenza vaccine induces IgE antibody production in the subjects, at the same time it also induced a higher IgG4 levels, and the IgG4 levels were actually higher than the IgE levels based on this data. And so the higher IgG4 levels than the IgE levels might explain why there was no anaphylaxis or there was no allergic reaction in none of the subjects. So then we compared the specific IgG4 IgE ratio between patients who develop anaphylaxis versus healthy subjects after influenza vaccination. And we observed that patients who develop anaphylaxis have a lower IgG4 IgE ratio as compared to healthy subjects. And so next we employed an ROC curve to determine if the IgG4 IgE ratio could serve as a biomarker for the diagnosis of patients with anaphylaxis. And so the ROC curve gave a higher sensitivity of 100% and specificity of about 95%, with an area under the curve to be 0.98 and a cutoff of less than 1.295. And so this data shows that patients with anaphylaxis So this data shows that patients who develop anaphylaxis after receiving the influenza vaccine have an impairment in the have an impairment in the production of IgG4 antibodies. And so, in summary, the hemagglutinin protein, which is the active ingredient in influenza vaccine, mediates influenza vaccine-associated anaphylaxis. And patients who develop anaphylaxis after the influenza vaccine have low levels of IgG4 antibody. And this IgG4 Ig ratio may serve as a biomarker for vaccine-induced anaphylaxis. And so the clinical implication of this study is that the design of influenza vaccine by altering the Ig binding epitopes on the hemagglutinin protein and concomitant induction of a potent IgG4 antibody production may prevent the occurrence of influenza vaccine-associated anaphylaxis. Thank you very much for your attention.
Thank you very much, Prince. That was like really nice talk, and the transitions. I see that some people have commented that they like the transitions of the slides. So yeah, it's great talk. So I can kick off the questions part by asking, do we know what contributes to this low levels of IgG4s in patients who eventually become anaphylactic when they receive the vaccine? Is there anything mechanistic, or are there hints that point to why they have this altered ratio compared to healthy people?
Okay, so patients with anaphylaxis, so the reason why they have low levels of IgG4 antibody might be due to their might be due to both their B cell regulatory and T cell regulatory immune system. So the T cell regulatory T cells actually mediates the production of IgG4 antibody, right? Mm-hmm. So it's possible that there is a defective in the T cell regulatory system in those patients. Mm-hmm. So I believe that might explain why they have low production of IgG4 antibody.
Okay, got it. Thank you for that. So Medina is asking, interesting talk, Prince. Thank you. How quickly does anaphylaxis develop in patients, and would a diagnostic allow an intervention before anaphylaxis happens?
When you receive a vaccine, so when you receive an influenza vaccine, so usually you are being told to wait for 15 minutes to assess for any potential allergic reaction, right? I think the minimum time for anaphylaxis to occur after influenza vaccination is 15 minutes, right? And with the biomarker, the biomarker, I believe the biomarker can only be used when, after there are signs of anaphylactic reaction. So when they see signs of anaphylactic reaction, then they can, collect your blood samples and immediately measure the IgG4 and Ig levels and determine the ratio. And so with that data, if the cutoff, so with that data, if you have a cutoff of less than 1.295, then then they can conclude that you are experiencing anaphylaxis with other signs of with other obvious signs of anaphylactic reaction.
Okay, got it. We have two more questions actually from our organizers. So the first is that IgG4 is only in the serum, correct? And if and then based on that, do you ever look at IgA to compare to IgE, or is it just IgG4 that you've looked at?
No, so we only look at IgG4 because the IgG4 is known to inhibit allergic reaction. Yeah, so like we never look into IgA.
Okay.
Yeah, and also I think like IgA is like I think IgA is a local antibody isotype. Yeah, so it's mostly so it's mostly found in the gut. Yeah, and not systemic.
Yeah, so it won't trigger probably systemic reaction.
No.
Yeah. Okay, the second question is great talk. Can the cytokine milieu explain the bias towards IgG4 instead of IgE? A cytokine drives the IgE type, so the immunoglobulin type. Have you observed a difference in vaccinated patients?
I saw in this study like we never evaluated the cytokine responses, but then again then again like interleukin 10 interleukin 10 is known to induce the production of IgG4 antibody, and so like with that we expect to see low levels of interleukin 10 in patients with anaphylaxis after receiving the influenza vaccine.
Okay, interesting. Okay, thank you very much. I think we've got to move on to our next speaker. So thank you very much, Prince, for this interesting talk and also for the questions and answers.
All right, thank you very much.
Thank you. So our next speaker is Dr. Sabelo Hadeba. So Dr. Sabelo is an immunologist by training and is based at the University of Cape Town in South Africa. He did his PhD in immunobiology at the University of Aberdeen in the UK, and is his first postdoctoral training at the Francis Crick Institute in London. He returned to South Africa after being awarded a Robert Bosch Fellowship in 2017. His main research focus is on immunology and genetics of allergic diseases. He uses the latest technologies to dissect complex immunological events in the context of homeostasis and disease. His future plan is to build a reverse translational program that combines basic science and translation at the human disease site. So we're excited to have you here. Thank you for accepting our invitation. And we're excited to hear your talk.
Thank you very much, and thank you for having me. And I hope that everyone is not like up early in the morning. For us here, it's in the afternoon or evening. So, and it's nice and warm because we're getting our summers here, and hope you're not too cold where you are. So I'm going to talk about one of the enzymes. It's called interleukin-4-induced gene 1. And it has a very interesting role in at least in type 2 immunity that we've noticed. And I guess it was sort of unexpected because it seems to be very tissue specific, which is why I called it yin yang, and you'll understand why I said that. And I have to thank Prince because I realized that in my slides I had not actually introduced allergic diseases, and I guess I assume that this session is really about allergic diseases, so I think everyone should be more or less familiar. But I think I'll touch on that. One of the things that I wanted to introduce you to is this enzyme called interleukin-4-induced gene 1. It's produced by many cells, for example, dendritic cells and macrophages, and in fact, it was initially discovered when B cells, human B cells, were stimulated with interleukin-4, and then they produce this enzyme. And also we know that it also has other functions in other cells, in adaptive cells, for example, Th17 cells produce it. It also seems to promote regulatory T cells. But it's not just the immune cells that produce the enzyme. You also find other immune cells that are able to do that. Spermatocytes, for example, and many others, they can produce the enzyme. As I mentioned, that the enzyme it has many functions. One of the things that has been shown is that it can inhibit activation of CD8 T cells, for example. So it inhibits their ability to secrete granzyme and interferon. And basically there's a cytotoxicity function. But it also can inhibit, or it can promote naive T cells to become regulatory T cells. So that's what other people have also shown. And what other people have also shown is that when it's produced by antigen-presenting cells, like macrophages, for example, its effect, particularly on the T cell synapse, so the T cell receptor interaction with an antigen-presenting cell, it can also inhibit that activation of T cells. So it has all these different functions. And so, oops, sorry, I've skipped a slide. I have skipped a slide. Let's see what has happened. Right, I was supposed to show you something, but it's not quite showing. It's underneath. You can probably see it. But what I was going to show you was that the enzyme can it likes these bulky amino acids, for example, phenylalanine and tryptophan. So it can take phenylalanine as a bulky amino acid, and it can digest it, and then it makes phenylpyruvate. That's one of the byproducts. But some of the byproducts that come from that are hydrogen peroxide and also ammonia. So it would break up phenylalanine, it makes up this byproduct. But when it takes tyrosine or tryptophan, for example, it will digest tryptophan, and it will then make these metabolites, and one of them is indole-3 aldehyde, or so you get many different metabolites. Some of those metabolites are kynurenine, but you also get indole-3 aldehyde. You also get many other metabolites that come from, I guess, catabolizing tryptophan into these different products. So so that's the slide that I was going to show you, but I think we still have it later, so you'll be able to see what I was actually talking about. So the work that I will present has been done mainly by these 3 or 4 students. Amgele did the bulk of the work for an MSc, and Nantobek is a PhD student, but she's done some of the work in this. And also Inam is a BSc honors student. She actually finished this year. And Fungai did a short postdoc with us before he moved to UPenn, I think. So some of the stuff that I present is really coming from them. So this enzyme is downstream of the interleukin-4 receptor alpha, as you all know that. Oh, somebody says fungi in my lab. Interesting. So it's downstream of the alpha receptor alpha. So you remember that RIS receptor binds both to interleukin-4 and interleukin-13. It's a heterodimer. So if you, for example, stimulate macrophages, these are macrophages, bone marrow-derived macrophages, you stimulate them with interleukin-4 or interleukin-13, then you look at expression of interleukin-4 induced gene 1. You can see that around about 10 hours, they peak if you stimulate with interleukin-4. So it seems that it's mostly interleukin-4 that's driving this. IL-13 does, but I think it's probably mostly interleukin-4. So what we then did, we generated a mouse that lacks interleukin-4 induced gene 1. And what we initially did here was that we took spleens from the same mice. We then sorted CD4 T cells, so naive CD4 T cells, and then we stimulate the CD4 T cells with either anti-CD3 or anti-CD28. And then we polarize them with using these different cytokines, either interleukin-12 and also inhibiting the other cytokines or interleukin-4. So you drive them to either Th1, Th2, or Th17s. And I think what was probably striking here is that if you look, for example, at GATA3 in the mouse that lacks the enzyme, you get an increase in GATA3, and also in terms of IL-4 production by the same cells. So you do see a bit of rho gamma T increase in the knockout mice, but this doesn't really translate to IL-17 production, for example. So what this was really telling us was that there seems to be if you polarize cells towards Th2, they are more likely to become, or in the knockout mice, they're more likely to become Th2 cells. So we then employed two models, which are models of type 2 immunity. The one model is asthma, so we use house dustmite to induce asthma in mice. The other model is a model of atopic dermatitis, and in this model we use it's a vitamin D analog called MC903. It induces a very acute itchy type of atopic dermatitis in mice. So if you shave mice and you apply it, it's going to cause itch. So let's look at the yang part of it. So this is the model of house dustmite. So you sensitize mice intratracheally, and then you come back, you challenge them over a 5-day period with the lower doses of house dustmite. You can then measure area hyperresponsiveness, and you look at other things. But what was interesting here, so this is a control mouse PBS. You can see here that it doesn't have any resistance, as you can imagine, because it's got no disease. And this is a wild type mouse, which is black. You can see that it does have a resistance. Also elastance, you can see it there. But in the knockout mouse, you do see a slight increase in terms of resistance, but it's not significant compared to the knockout. But in terms of elastance, you do see a slight increase in the knockout mouse, which probably confirms partly what we are seeing with the Th2 polarized cells in vitro. But if you then go and look at things like mucus production, so this is mucus production, this is the goblet cells, you can see that on pus staining, there isn't really much of a difference between the two mice, although it seems that it's slightly lower in the knockout, but it's not really significant. But some of the things that we found to be striking was an increase in eosinophils, for example. So you can see here that if you this is the knockout mouse and you compare it to a wild type and you've sensitized them with house dustmite, an increase in eosinophils, no real change in either in total cells or neutrophils. So it seems to be specific to Th2. And then we look at cytokines. So this is stimulating cytokines which anti-CD3, and then you measure Th2 cytokines in supernatant in the lymph nodes. So you can see that even the Th2 cytokines are increased in the knockout mouse that are sensitized to house dustmite, whether you look at IL-13 or IL-5 or IL-33. This is in the total lung. So you also see that this increase in Th2 cytokines. So one of the things that we're also interested in was what are the cells that are actually producing these type 2 cytokines. And some of the things that we did find out were generally ILC2s are increased, but we also see that even the production of type 2 cytokines by ILCs is also increased in the knockout. But you also do see an increase in even by CD4 T cells So which sort of confirms what we had seen in terms of the increase in eosinophils in the lung, but also what we had initially seen in vitro. But what is really interesting is that if you now look at B cells, so you're looking at total IgE, which you'd expect to be increased, it's actually not, it is decreased. Also antigen-specific or house dust mite-specific IgE is also decreased. So if you compare, this is a wild type and this is a knockout, you can see that that is decreased and these are just controls. But also we can confirm that even class switching doesn't seem to be happening in the lymph nodes, because you also see a decrease in the number of germinal centers. So it seems that although in the lung you have this aberrant Th2 increase in the knockout, but actually in terms of IgE, that doesn't seem to follow suit. So we tried many different things to try and understand and to also dissect, what was probably going on. And one of the things that we eventually did was to take bone marrow derived dendritic cells that we had in vitro, that we generated in vitro with GM-CSF. And we then passed these dendritic cells with house dust mite, and then we give them to the mice at day 0. And then we come back and challenge again with a lower dose of house dust mite. And then we can also measure these parameters like area hyperresponsiveness and cytokines. I'll just show you some of the data here. So what you are seeing here is so this is looking at eosinophils, which is what we had seen being increased in the mice. So this is looking at mice. So these are wild type mice that we've given wild type dendritic cells, which are here. And this is knockout mice that we've given wild type dendritic cells, and you can see that that's pretty similar. But if you give the knockout mouse knockout dendritic cells, so you can see that you still have this increase in eosinophils, which confirms that, you have this aberrant Th2 immunity in the absence of the enzyme. If you look at, for example, ILC2s that are producing IL-5 or IL-13, again, if you give the knockout mice dendritic cells from knockouts, they seem to have this increase in type 2 immunity. But if you give the knockout the wild type dendritic cells, there is this reduction in Th2. So what it's telling us is that these dendritic cells can suppress this aberrant Th2 immunity that you find in the knockout. So I've told you that the L4, in the absence of the L4R1, you get this increase in Th2 immunity, although you do see a decrease in IgE and class switching. But actually, what I'm going to show you now is a complete 180-degree turn. I mean, it's a complete opposite. So in this atopic dermatitis model, so this is how we model atopic dermatitis. We use MC903. We don't always use the same dose because it's a bit it's not very stable. So you sometimes have to change the dose a little bit. And what we do is we shave the mouse and we apply it topically until day 9, and then you sacrifice at day 10. So all that it does, it causes this acute itch. And also the features that you see are pretty much similar to what you'd see in dermatitis. And one of the ways in which we can see that is that if you give the mice MC903, they lose a little bit of weight. So these are wild type mouse. Around about day 5, they start losing a little bit of weight. So no, it's not much, but they do lose a little bit, about 5 to 10%. But if you look at the knockout, so this is the knockout here, which is the black triangle. It's not really losing any weight. This is just our controls of ethanol, because we dilute this in ethanol. So obviously they don't lose any weight, but so that was interesting. Similarly to what we had seen with the asthma model, we still see this decrease in total IgE in the knockout mouse. So that at least was consistent. But then if you look at things like, now this is looking at epidermal thickness. I'm sorry that my slides look weird. They seem to have changed a little bit. So this is ethanol controls in the wild type and the knockout. So wild type is at the top. So you can see that in the wild type, you can see this thickened epidermal layer, and it's less so in the knockout. And you quantify that here, you can see that the knockout has less epidermal thickening compared to the knockout. Sorry, compared to the wild type. And if you then look at this is Th2 cytokines in serum, you can see that there's a reduction in things like TSLP, a slight reduction in IL-4, and also a reduction in IL-5. In the mice that have been given MC903, in the knockouts, you see that this is reduced compared to the wild type. So this is actually what I wanted to show you initially, that this is tryptophan here at the top, and this is interleukin-4 induced gene 1, and it can catabolize tryptophan to have these different metabolites, one of them being caronerin, and the other ones being indole-3 pyruvic acid, which also can further be broken down to these different types of metabolites. So what we wanted to know here was that if you remove the enzyme, because essentially this is what we are seeing in atopic dermatitis, is that you remove the enzyme, you have reduced disease. So if you bypass the enzyme and you add some of these metabolites back, would you be able to induce the disease? And that's essentially what we did here, is that we sensitized the mice with MC903, and then we give some of these metabolites back. So the one that we've started with is L-carnitine and also indole-3 aldehyde. So we treat with these topically also pretty much every day. And what you are seeing, I'm sorry that these are not exactly perfect as they should, but you can see here, this is ethanol, this is epidermal thickening that you'd see, and you treat with MC903, this is the epidermal thickening that you see, and also all the knockout and the different treatments. So we've only added these metabolites in the knockout mice. So you can see, for example, here, this is the ethanol, this is the amount of epidermal thickness that you see, and this is what you see in the wild type. When you treat with MC903, it increases, and the knockout decreases, which is what we had seen before. And if you then add either indole-3 aldehyde or L-carnitine, you restore this epidermal thickening, which is very exciting, because it essentially tells us that interleukin-4 induced gene 1 is upstream of these metabolites, and it potentially is catabolizing tryptophan, and if you restore the function of the enzyme by going downstream and putting back these metabolites, you are able to restore the function. And this is something similar we see with total IgE. This is what you see in the wild type, and in the knockout you see reduced IgE. As you'd expect, you add at least L-carnitine, it's able to restore the total IgE. It's probably similar with, for example, other Th2 cytokines and even interferon gamma. That seems to be, the addition of the metabolite seems to restore the function of interleukin-4 induced gene 1. And so our working hypothesis at the moment, at least for the asthma model, is that there is a particular cell that is secreting the enzyme, and we think these are antigen-presenting cells, probably, as we've shown, that it's dendritic cells. And what they are able to do is that they are able to inhibit the production of Th2 cytokines by either ILC2s or TLP2 cells, and that's how they dampen the Th2 immune response. Although what we are probably seeing in atopic dermatitis is a complete opposite, where we think that the enzyme catabolizes tryptophan, for example, and then produces these metabolites. These metabolites probably act, we know that they are ligands for aryl hydrocarbon, which senses these metabolites, and probably we think that this is the reason why you have this dichotomy in terms of either suppressing Th2 immunity or promoting Th2 immunity. And this is something that seems to be very specific in a particular tissue, as we've shown that in the lung it does something different, and in the skin it does something different. With that, so these are some of the stuff that we're actually looking at now a little bit more, and I think we'll probably want to really follow up on how does alpha L1 regulate Th2 immunity. And we're starting now to look at the potential diagnosis of this, and also how do we therapeutically target alpha L1 in atopic dermatitis. So I'm going to thank everyone, and particularly the students that have done most of the work, and also Division of Dermatology that are helping us with the access to patients. Thank you.
Thank you very much, Sabelo. That was really great. It was a really fantastic talk and really exciting data. Thank you for sharing actually unpublished data, which is really nice to see here. So we are waiting for questions, but while we're waiting, I can also exploit my position now that I'm chairing this and ask the first question. Have you tried an infection model that is like more Th2 inducing, like NIPU? So of course, everyone does NIPU now. And then have you compared that with the other type, which is like a Th1 inducing, like, I don't know, toxoplasma or something?
So what we have tried in the lab is we've tried Mycobacterium tuberculosis, and in that particular case, it seems to, because the enzyme is also involved in polarizing M2 macrophages, and if you remove the enzyme, then the macrophages are able to kill better Mycobacterium. So it seems to be, you actually get reduced CFUs when you do that. We haven't really tried NIPO, but we have tried, I think some people have tried Schisto, but there wasn't really any effect. And I think it may be due to the fact that it's, it probably plays very different roles depending on whether it's acting on regulatory T cells, for example, or it's suppressing Th2. So, and some of these models, they will go through phases of switching from Th1 to Th2 or to Treg.
Yeah.
So it probably is part of the reason why we haven't. But yeah, we've tried other models, for example, that have a mix where we actually expected to see an effect. For example, Leishmania is very much Th1, and you would expect that if you have this downregulation of Th2 or even a hyper Th2, that the mice would be more susceptible, but that's not the case. You do see a general increase in IgE, but it doesn't translate to, quicker elimination of parasites, for example. But I think it's very specific also, because if you do that if you take out macrophages, for example, and you infect with a parasite, you see an effect. But if you in a complete mouse in vivo, it doesn't translate to that. So I think it probably is something that's it's probably also very localized in specific cells in terms of how it functions, but maybe that gets masked in some ways in a complete model.
Yeah. Great. We have a question here from Professor Walker. Fascinating talk. Thank you. Are there SNPs in the IL-4 inducing one gene? Are these associated with asthma or atopic dermatitis?
Thank you, Prof. Walker, for that question. So interesting. So last year, I spent some time in Bristol in epidemiology or in statistical genetics lab, And we've actually noticed SNPs that are in alpha-1 gene in atopic dermatitis. We haven't really looked in asthma, although it doesn't probably fit the category, 5 times 10 to the 8 that you'd want to expect, or p-value that you'd expect for any SNP. So it doesn't pass that, but it's somewhere around. I think 1 times 10 to the 4. So it is associated with asthma, but it's not. It doesn't probably reach the cutoff that you'd expect for those kind of studies.
Yeah.
But I mean, we are trying to look at for example, in asthma. So we that's one of the things that I'm supposed to be doing. But yeah, we'll see. It will be interesting to see. But I mean, the problem also with SNPs is that even if you get it, sometimes it doesn't actually tell you functionally what it does. Is it a protective SNP, or is it a pathogenic SNP? So I think those are some of the things that we probably need to probe.
Yeah, exactly. Yeah. Thank you very much. I think it's now time. So I think, we are on time. So thank you very much again, Sabelo, for this nice talk and for the nice discussion. I think it's now time to move on to our last speaker of this session. So our last speaker of the session is Dr. Evonne Jean. So Evonne is a fifth-year PhD student, a candidate in the Dabrowski-Herbert Lab, completing their dissertation work within the IgG program at the University of Pennsylvania. Having completed their Bachelor's of Science at the illustrious Hampton University in 2019, with a focus on molecular and cellular biology, Evonne entered their PhD with an interest in studying protective immune responses at the cutaneous barrier. While studying a historically overlooked initial stage of infection of parasitic worms in immunology, which is skin penetration, Evonne developed a novel model to investigate protection against percutaneous penetration, which was previously unexplored. Evonne enjoys reading and writing and hopes to implement these skills into a long-lasting career in science communication. Following their PhD training, outside of the lab, Evonne is a strong advocate for diversity and inclusion in academic science. So we are excited to have you here, and take it away. Thank you very much.
Thanks. I hope everyone can hear me and see my screen. I am Evonne. I'm a fifth year at Penn, and my talk today is titled IL-33 is required to mediate gamma delta T cell dependent resistance to skin penetrating helminths. So by way of introduction, I wanted to start off by introducing my lab. The Dabrowski-Herbert Lab, this is my PI, and this is our lab webpage. Fungi, who was in the lab of the last speaker, is actually in our lab now, doing amazing work. He's really the life of the party there. I'm really glad that you guys can let us have him. And so I wanted to talk about what we do at the Herbert Lab. In general, our lab is interested in understanding protective immune responses against helminths or skin-penetrating gastrointestinal nematodes. These worms are an excellent tool to study type 2 immunity, and we have a number of different projects in the lab as illustrated by this cartoon, which we use this model as a model of host-parasite interactions and the mounting of adaptive or acquired immunity. And so these include projects such as our ongoing inquiry into the role of IL-33 and mediating protective immune responses against worms, which I will touch on in my talk, and also the role of IL-33 in mediating acquired immunity by way of activation of lymphocytes such as Th2 or T helper T cells and gamma delta T cells. So if we specifically look at diseases across the globe or infections that require type 2 immune responses, we see this inverse relationship between regions of the world where there's a prevalence of allergy and asthma as compared to where there's a prevalence of helminth infections and disease. And so I really want to emphasize here that both allergic and asthmatic responses and helminths require a similar induction of type 2 immunity. Whereas is the case in helminth infection, a type 2 immune response is required for resolution of infection. In the case of allergy and asthma, the type 2 immune response can sometimes be inappropriate and significantly decrease the quality of life in individuals with these conditions. Therefore, studying the resolution of type 2 immunity in our lab using helmets, using soil-transmitted skin-penetrating gastrointestinal nematodes is imperative to both the developed as well as the resource-deprived nations of the world. While we have a number of species of helminths in the lab, such as Nipostrongylus, Schistosoma, and Trichuris at one point, and Strongyloides, which I work with, Strongyloides rats in particular is a soil-transmitted skin-penetrating gastrointestinal nematode, or helminth, and this slide shows the prevalence of infections by this particular worm across the globe. And helminths infect over a third of the global population, and although they have low mortality, they present a number of morbidities, including anemia, diarrhea, as well as cognitive impairment in children, as well as increased risk of co-infections with other endemic pathogens, such as malaria and HIV. So these morbidities significantly decrease the quality of life, and although treatments do exist to combat morbidities in helminth infection, these treatments do not promote a robust immune response in people, leaving humans susceptible to reinfection with multiple worms. Unlike our mouse model systems, where mice do develop a robust secondary immune response, secondary type 2 immune response against reinfection. In general, the life cycle of most soil-transmitted gastrointestinal nematodes is as follows. So worms start off as eggs in the external environment, developing to the first and second larval stage in the soil. Once they reach this third larval stage, they become infectious and penetrate the skin of hosts in order to establish infection. Post-penetration, they disseminate quickly through the bloodstream to reach the head and the lungs. And here in the lungs, they develop into a fourth larval stage before being coughed up and swallowed in order to enter the intestines. Here in the intestines, the worms mature into adult worms and can sexually reproduce eggs, which are expelled in the feces of the hosts. And this reestablishes the life cycle in the external environment. While most studies have focused on protective immune responses at mucosal barrier sites, such as the lung and gut, I'm most interested in this initial stage of infection, which is percutaneous penetration and cutaneous helminth infection, as this area of study has been severely overlooked in anti-helminth investigations. Clinically, helminths in the skin have been documented to elicit strong inflammatory responses, as seen in this condition known as cutaneous larval migrans. And so this condition can be resolved naturally in people within 48 weeks, although these studies have been unable to show whether this condition is a result of larvae trapping in the skin to kill the parasite, or whether this is just a result of an inflammatory response from larvae traveling through and away from the skin barrier. If we dive down to the skin microenvironment, we can see it's a complex mosaic of these hematopoietic cells in the dermis, as well as epithelial cells in the epidermis, very structured in the way they are formed. And this skin immune axis has been studied in a number of contexts, such as fungal and bacterial infections. However, how the skin immune axis changes to prevent or create acquired immune response against cutaneous helminth infection is poorly understood. Most experimental systems that investigate anti-helminth immunity bypass the epidermis during parasite inoculation to administer the parasites directly into the subcutaneous space. So this leaves speculation as to whether the upper layers of skin are involved in protective immune responses. And so my thesis project involved a new strategy where worms must burrow and penetrate all layers of the skin in order to test the mechanisms of a host protective immunity and determine whether immunity is mounted against larval penetration. This cartoon summarizes what has been elucidated thus far about protective immune responses in the skin to helminth infection and models such as Nipostrongylus and Strongyloides. It's much appreciated the roles of neutrophils and eosinophils and expunging DNA and histone-coated nets can trap and kill the parasites at the skin barrier as well as other sites in the body. But very few investigations have shown an acquired immune response that can trap these worms. There is studies that have shown that M2 macrophages mediate secondary immune responses in a clear type 2 immunophenotyped response that required IL-4 from basophils in a Nipostrongylus model. And another paper showed that eosinophils and neutrophils are primed to efficiently and rapidly kill parasites in a secondary infection at the skin. However, these papers did not show any roles for antigen-presenting cells such as dendritic cells or lymphocytes such as LC2s and 8-2 lymphocytes, or CD4 positive Th2s or T helper cells, or skin resident gamma delta T cells, which are resident both in the skin as well as the gut and lung. Additionally, these papers also had the caveat of using the common injection model to directly inject the worms into the subcutaneous space and bypass the epidermis and mediating any involvement in protective immunity. These papers also couldn't ask whether there's acquired immunity against penetration by worms. So therefore, in my research question, I'm asking, would a natural route of infection change the immunological mechanisms that are involved in protective immunity? And if so, can we elucidate what those mechanisms are? In order to study the protective immunity against larval entry, I developed the following model in which mice are sedated under ketamine xylazine, and the hind paw is submerged in a certain inoculum of Strongyloides vaccae larvae, or IL-3. This allows the larvae free movement to naturally penetrate the mouse skin over 40 minutes of exposure to the worm solution. In order to test whether acquired immunity is mounted against penetration, I've used the following timeline where mice are infected at day 0 with the primary inoculum, quantifying the number of non-penetrating worms after 40 minutes in order to assess the penetration efficiency of the inoculum. 12 days post-primary infection, the mice are reintroduced to a secondary infection, again quantifying the number of non-penetrating worms after 40 minutes. And 2 days post-secondary infection, the mice are euthanized in order to assess the mounting of an inflammatory response in the skin as well as the proximal palpable lymph node. Using this model, I was able to generate the following data, in which you could see there's a significant decrease in the penetration efficiency of these IL-3 larvae in a secondary infection as compared to the primary infection. And so this data is indicating that wild-type mice are able to resist or develop an acquired immune response against penetration by these worms. We also confirmed this by taking head counts of the number of worms that are in the head at 2 days post-infection. And you can see that there is a substantial decrease in the number of worms that are able to disseminate away from the skin and reach the head and a secondary as compared to the primary infection at 2 days post-primary or 2 days post-secondary. So with this model, I'll be investigating the hypothesis that acquired resistance to percutaneous infection requires type 2 lymphocytes and a type 2 immune response requiring these CD4 helper cells as well as resident gamma delta T cells in the skin. So in order to investigate the requirement of type 2 immune responses, we turn to STAT6, as STAT6 controls a number of functions mounted in a type 2 immune response that's been shown in the lung and gut to be required for anti-helminth immunity. So these functions include things such as polarization of empty macrophages, which I previously told you was shown to be required in anti-helminth immunity in the skin. STAT6 also can help polarize and activate Th2 and ILC2, these being our lymphocytes, allowing them to create or make type 2 cytokines, such as those listed here. STAT6 is also implicated in the physiological responses that are required for expelling helminths from the gut, including smooth muscle contractility, as well as mucus production by goblet cells in the gut. And so this is part of the weep and sweep response that expels helminths from the intestines. So we wanted to see if STAT6 is required in my model. And so we used these STAT6 knockout mice where we infected STAT6 knockout mice or wild-type mice with a primary secondary infection. Surprisingly, we found that STAT6 is not required for acquired immunity. As shown by the data, STAT6 knockout mice were just as efficient as the wild-type mice in acquiring resistance against penetration. So despite these surprising results, I wanted to further investigate these mice to determine what immune functions were still intact in the absence of STAT6, which could be mediating acquired immunity. And so I did that through flow cytometry in the skin using the gating strategy on the left here. So we identified CD4 positive T cells from the CD3 positive population, and the Th2s were designated by expression of GATA3. And as you can see, there's an expected reduction in Th2 cells in the absence of STAT6. And we saw similar results with ILC2s from the double negative population. However, when we investigated gamma delta T cells through the CD4 negative, TCR delta positive T cells, we didn't see any differences in the number of gamma delta T cells in the skin. So could gamma delta T cells be meeting immunity in the absence of STAT6? we'll get to that at the end. And we saw similar results in the popliteal lymph node, which is proximal to the foot pad. So I wasn't too eager to give up on the idea of type 2 immunity being required in this model. And so going back to the literature, we also have seen that IL-33 can promote type 2 immunity independently of STAT6. So IL-33 is an alarming cytokine canonically released by epithelial cells in the context of damage induced by helminths. And IL-33 induces type 2 immunity through activating type 2 lymphocytes in myeloid cells by recognition of its receptor ST2. And so I next wanted to ask the question, could IL-33 be mediating anti-helminth immunity in the absence of STAT6? And so to answer this question, we use a similar approach where we have these IL-33 deficient mice. So the CMV-CRE driver will allow for these mice to be completely deficient IL-33 expression. And we saw that as compared to our wild-type controls, these mice were unable to reduce the penetration efficiency of these Strongyloides rati infectious larvae in a secondary infection as compared to primary infection, indicating that these mice are unable to acquire resistance in the absence of IL-33. We saw similar results when we blocked IL-33 signaling by blocking the ST2 receptor, which I don't have time to show right now. But to investigate the immune repertoire of these cells in a similar way to the STAT6, we saw that there is a significant reduction in the number of gamma delta T cells, and particularly the IL-33 responsive gamma delta T cells, which express ST2, indicating that IL-33 could be required to induce expansion of these gamma delta T cell subsets. And we also saw no differences in the CD4 T cells. So it seems to be that the gamma delta T cells might be the most important in this particular model to promote acquired resistance against penetration. And so I wanted to bring you to this model so we can summarize what I've shown you so far. There's just one more experiment after this. So far I've shown that IL-33 canonically released by epithelial cells in the context of damage allows for the expansion of gamma delta T cells. But what I haven't shown you is, are gamma delta T cells required for acquired immunity, or is this just some offhand effect? Because there's a lot of other cells that could be at play here. And so to determine whether or not gamma delta T cells are required, we again performed a knockout experiment using these TCR delta gamma delta knockout mice. So these mice lack all gamma delta T cells. And we saw similar results where there's an inability to reduce the penetration efficiency, indicating that these mice are unable to acquire resistance against penetration and secondary infection as compared to primary infection compared to our wild-type controls. And so this leads me to my model, where I think IL-33 is required to induce gamma delta T cell populations at the skin barrier site to promote some change in the skin barrier that allows for repulsion of worms in a secondary infection. And data I don't have time to show. We did do a lot of H&E staining of the skin to see what, because I originally thought that the maybe the skin barrier was thicker and that could be relating to repulsion of the worms, but we didn't see any differences really in thickness of the barrier. So we next hypothesized that there's an increase in skin barrier integrity that may be mediated by gamma delta T cells, as gamma delta T cells have been reported in the literature to be important for homeostasis and repair from injury of the skin. And so with that, I'm just going to take you to the model. I don't have time to show this data, but we did see differences in these ECM or extracellular matrix and adhesion molecules genes at the skin, which we think may be increasing the skin barrier integrity. And so next, we want to test whether or not these increase in skin barrier integrity genes are a direct result of gamma delta T cells, and also more specifically, what are the functional consequences of IL-33 engagement with gamma delta T cells. And I'm actually performing these experiments right now. Hopefully we'll have results in the next 2 weeks, and I'll be able to answer these questions and then move on and really complete this model. And so with that, I'll take it to my acknowledgements. I want to acknowledge my lab, the best lab in the world, of course, and everyone who helped me with this, including my thesis committee and IgG leadership, IgG being the immunology graduate group at UPenn, my funding, and then obviously Black and Immuno for allowing me to come and speak on this panel. So with that, I'll take questions if there are any.
Thank you very much, Evonne. That was really nice. It was really great talk and fascinating. Yeah, a lot of interesting things. I never knew that actually gamma delta T cells can express ST2, which is interesting. So questions. Let's see if there are any. But if there are not any questions, so I can ask the questions while the others are preparing them. So basically, you need gamma delta T cells for this initial protection. Do we think that some cytokines are important for that? I mean, are gamma delta T cells, for instance, directly killing the parasites, or is it like they just non-canonically, let's say, contribute to the thickness, let's say, of the barrier?
Yeah, so I don't see any differences in the thickness of the barrier. And I don't know about killing parasites. So the investigation is more of a stopping of them from even infecting the mouse in the first place. So if they can't get into the skin, then they can't infect the mouse. And I think that there's a prevention of penetration as opposed to them killing the worms directly. Gamma delta T cells are known to make a lot of different cytokines, and we investigated a few of them, such as IL-17A, IL-22, TNF alpha, interferon gamma, AREG. And so I do think there's a difference in AREG, which AREG would be a molecule that would help the skin barrier increase its integrity or repair from injury. But I'm not sure about the other cytokines. Yeah, I can't really make a conclusion one way or the other. I've done a number of experiments, but I don't really want to make a conclusion on that just yet.
Okay. And Sabelo actually asked the follow-up question, is that whether they make IL-4 after activation?
Yeah, so I don't think that gamma T cells make IL-4. I think they're more like Th17. So if you think of gamma T cells as more of a Th17 phenotype, I don't think they lean towards type 2 cytokine production, at least from my studies. They do seem to maybe be a small population of IL-13 positive gamma T cells, which we're going to investigate in my next experiment. But yeah, so we'll be looking at that more closely. But I don't think they make IL-4.
Okay. I have another question, and there is also another question. So the question I'm asking, whether you've looked at CD4s in general, but do you think that subsets of CD4, I'm specifically talking about Tregs, can be like in any way, let's say, reduced in your model by, or they are important, not affected by STAT6?
Yeah, I don't see any differences in Tregs. I can't remember the STAT6 data, but there is probably a difference in the GAD3 positive Tregs. But overall Tregs, I don't see a difference really in the absence of IL-33 per se.
Okay.
I think it's more like the functional differences as opposed to the overall, are the cells there?
Okay. Sabelo said that he remembers a paper on gamma delta T cells in skin cancer surveillance through IgE. So that might be of interest. Medina is asking, how long do you think this acquired immunity lasts? Have you looked at secondary challenge beyond 12 days?
Yeah, so this is a question I actually get a lot. And I'm planning to experiment, like a long-term experiment with some mice that I have soon about that. I'm hoping it's a long-term immunity, but I can't say for certain. Based on how short the timeline of my experiments is, I'm really just not sure. It's a short timeline of my experiments.
Okay. Yeah, it would be interesting to see if it's like really a memory response or something. Okay, let me scan again and see if there are more questions. I think there are no more questions. So thank you very much, Evonne, for this interesting talk. And thank you actually for all of our speakers for sharing the work, and for the attendees for the actually nice discussion. And we hope actually that you will stick around for the next panel, the panel discussion that we have, which is on AI machine learning in immunology. So stay tuned, and thank you very much.